Hello everyone. Today I will present about our research on the alteration of microflora and antimicrobial peptides in the oral cavity of bee pollen-fed mice. In the following, I will explain how the research was conducted and the results. Bee pollen. Various products of bees have been used as natural remedies since ancient times. The wide use of these products is due to broad functions such as anti-inflammatory, antibacterial, and many other properties. Among various bee products, bee pollen has been studied and used mainly due to its antioxidant property, which has been attributed to its polyphenolic component. The increasing interest in bee pollen for both its value as a diet supplement and certain potential curative properties emphasizes the need for further research. Bee pollen and microbiome. Microflora plays an important role in regulating the immune system through host-microbe interaction. Although few studies have focused on antimicrobial effect of bee pollen, concrete evidence on how bee pollen affects microbial community of oral cavity is lacking. The metagenomic study of oral cavity and intestine might be helpful in elucidating the effect of bee pollen on microbiome. Antimicrobial peptides, such as beta defensins, are the major defense system in maintaining the balance between microbes. The study of AMPs could help in understanding their role in antimicrobial study of bee pollen. Objectives. We have two objectives for our experiment. First, to perform metagenomic analysis of oral microbiome of mice fed with bee pollen diet and normal diet. Second, we will perform QRT-PCR to check for relative mRNA expression of common antimicrobial peptides. Materials and methods. We started this research by checking whether bee pollen has effect on new oral bacteria. For this, minimum inhibitory concentration of Streptococcus mutans and Porphyromonas gingivalis were determined in vitro, and the following is the details. To determine the effect of bee pollen diet in vivo, 6 weeks old C57BL/6J mice were randomly divided into two groups, bee pollen diet group and control group. Experiment group was fed by 5% of bee pollen food, while control group was fed normal mouse food. After 1 month, mice were sacrificed for collecting oral microbiome and buccal mucosa. Metagenomics of oral microflora. We collected saliva by swab and kept it in TE buffer. The oral microbiome was extracted using lysozyme and DNA easy blood and tissue kit from Quaggan. The first step in metagenomic sequencing is the applicant PCR, which targets V3, V4 region of bacteria 16S ribosomal RNA. In second step, we did index PCR to tag all the amplified DNA. Then thirdly, library quantification, normalization, and pooling was done, and then library denaturing and MESEC sample loading were performed. Data analysis. The data of metagenomic sequencing was analyzed with quantitative insights into microbial ecology too. Diversities were analyzed as alpha diversity and beta diversity. mRNA analysis of antimicrobial peptides. RNA was extracted from the buccal mucosa, reverse transcription done, and QRT-PCR was performed. Mouse beta defensin 1, 2, and 3, cathelicidin, and S100A8 and S100A9 were checked. Results. The MIC results showed that there was no growth of Porphyromonas gingivalis at bee pollen concentration of 2.5 to 10%. Therefore, 2.5% was taken to be the MIC of bee pollen for Porphyromonas gingivalis. Taxonomy plot of oral microbiome. Here, we can see visible alteration in number of some microflora. The yellow bar represents the relative abundance of bacterial Lactococcus at genus level. The ANCOM analysis showed bacterial Lactococcus was significantly increased in bee pollen group as compared to control. The alpha diversity as measured by Shannon group significance of oral microbiome in bee pollen group was significantly reduced. The beta diversity as measured by weighted unifrac of oral microbiome in bee pollen group was significantly altered. The mRNA level of defensin B1, defensin B2, defensin B3, cramp, S100A8, S100A9 were evaluated in the buccal mucosa. Mouse defensin beta 2 and 3 were significantly upregulated, while cathelicidin was significantly downregulated in bee pollen diet group as compared to the control. Discussion. Lactococcus at genus level showed increased relative abundance in the oral cavity of bee pollen-fed mice. Lactococcus is a gram-positive anaerobe and is a probiotic with beneficial characteristics for human health, including reduced serum cholesterol level, improved balance of intestinal microflora, immunomodulatory activity, and skin health. The increased proportion of Lactococcus in oral microflora may have a beneficial effect on systemic as well as oral health. Lactococcus has an inhibitory effect on Streptococcus mutans and prevents biofilm formation containing Porphyromonas gingivalis. Therefore, bee pollen may have beneficial effect on oral health via increased proportion of Lactococcus. Although no antimicrobial activities of bee pollen against Streptococcus mutans was found, bee pollen inhibited the growth of Porphyromonas gingivalis. Bee pollen contains certain reducing and non-reducing sugars. Streptococcus mutants might grow using low sugar in bee pollen, resulting in no inhibitory effect of bee pollen on the bacteria. However, Porphyromonas gingivalis is an ascarolytic anaerobic bacteria, and low sugars cannot be utilized for their growth. The significant increase in beta defensin 2 and 3 and reduction of cathelicidin in the oral cavity of bee pollen-fed mice could be a cause or result of alteration in the relative abundances of bacteria. Further studies are needed to understand the complete mechanism. Conclusion. The bee pollen diet altered the microflora and antimicrobial peptides of oral cavity. This finding signified that bee pollen could modulate the immune system by altering the host-microbe interaction. Thank you.